What if I told you that some organisms we've long studied as fungi aren't actually fungi at all?
Hmm.
Today on the Deep Dive, we're peeling back the layers on a truly fascinating biological mystery, diving into kingdom Straminipila.
Yeah, this group, it's traditionally found its home in mycology textbooks, like the chapter we're pulling from Introduction to Fungi.
Right, but modern science shows they have some incredibly distinctive traits, things that set them really far apart from true fungi.
That's exactly it. It's a classic case of convergent evolution, perhaps, where different organisms evolve similar lifestyles.
So it looks apart, maybe acts apart.
Exactly. While Straminipila might look and act like fungi, sometimes many are decomposers or parasites. Their fundamental cellular makeup, their biochemistry, it tells a completely different evolutionary story.
And understanding these differences isn't just, a nerdy scientific detail.
Not at all. It reshapes our whole understanding of life's diversity. And it reveals some pretty surprising impacts on our world.
Okay, so our mission today, uncover the crucial biological blueprints that define the Straminipila. We'll delve into their signature hair-like bits, the flagella that actually give the kingdom its name.
That's a key part.
And then we'll explore two specific phyla within this kingdom, the Hyphochytriomycota and the Labyrinthulomycota.
Right, looking at their unique life cycles, their ecological roles.
And some really unexpected real-world significance. Get ready for some aha moments, because this is where our picture of life's family tree gets, well, seriously interesting.
Definitely.
Okay, let's unpack this biological puzzle. The kingdom Chromista, now more widely known as Straminipila. It was set up specifically for these eukaryotic organisms that just didn't quite fit elsewhere, right?
That's right. Even if they shared some superficial similarities with, say, protozoa or fungi, it was clear they needed their own grouping.
Like finding a new, unique wing in the grand mansion of life.
Sort of, yeah. The question was, what were those undeniable differences that forced this reclassification?
Okay, lay it on us. What's the blueprint?
Well, the scientific community zeroed in on 5 crucial characteristics. Think of them as fundamental design specs that clearly distinguish Straminipila from true fungi, the Eumycota, and other kingdoms.
5 key things. Got it.
First up, cell walls. Unlike the chitin you find in true fungal cell walls.
Right. Chitin like in insect shells.
Exactly the same stuff. Straminipila. Their cell walls are primarily made of cellulose.
Cellulose, like plant.
Precisely. Like plants, that immediately tells you, okay, this is something different.
Wow, okay. So cellulose wall, big red flag number 1. What else?
Second, their mitochondria. The powerhouses of the cell.
Sure.
Inside them, the inner membrane folds into structures called cristae. In Straminipila, these cristae are tubular, like tiny distinct tubes.
Tubular cristae. Okay, how's that different?
Well, in true fungi and animals, the cristae are usually lamellate, more like flattened plates or shelves.
Ah, I see. Tubes versus plates. Another fundamental difference in the internal machinery.
Exactly. Points to very different evolutionary paths.
Fascinating. Even energy production is distinct. What's number 3?
Third is their Golgi apparatus. These are the cell's processing and packaging centers for proteins.
Right, the cell's post office.
Kind of, yeah. Straminipila have prominent, well-developed Golgi stacks, sometimes called dictyosomes. In true fungi, the Golgi is typically much more reduced, often just single, flattened sacs or cisternae.
So a more complex packaging system is Straminipila.
Suggests maybe more complex processing or secretion going on, yeah?
Okay. And the 4th characteristic. You mentioned it's literally in the name.
It is. Many Straminipila have flagella whip-like tails for movement at some point in their life. But here's the kicker. They always include a unique type called a Straminipilous flagellum.
The namesake.
The namesake. It's so diagnostic, so unique, that it led to renaming the whole kingdom. We'll definitely dive deeper into this signature flagellum in a bit because it's quite a marvel.
Okay, can't wait for that. That sounds like strong evidence for a unique group. What's the 5th and final piece of this biological blueprint?
The 5th one is biochemical. It's about how they make an essential amino acid, lysine.
Lysine synthesis, okay.
Straminipila synthesize lysine using the alpha-epsilon diaminopimelic acid pathway, or DAP pathway for short.
DAP pathway. Does that ring any bells elsewhere?
It does. It's the same pathway found in plants, green algae, and even bacteria.
Whoa. Okay, so not like fungi at all.
Not at all. True fungi almost exclusively use a different pathway, the alpha-aminoadipic acid pathway, or AAA pathway.
So it's like a molecular fingerprint.
It really is. It tells us these organisms share a deep, fundamental biochemical ancestry with plants and bacteria, which is pretty mind-blowing when you consider how some of them look or act like fungi.
Okay, let's pull this together. Cellulose walls, tubular mitochondrial cristae, prominent Golgi, that signature straminipilous flagellum, and the DIP pathway for lysine. That's a powerful set of characteristics.
It's a very clear biological blueprint, yeah.
And it clearly separates Straminipila from true fungi, even if groups like the Umycota, Hyphochytriomycota, and Labyrinthulomycota were traditionally studied by mycologists just because of, well, how they lived.
Right. Lifestyle similarities confuse things for a long time.
And crucially, this separation into one group, meaning they all share a common ancestor, they're monophyletic, that's been backed up by genetics, right?
Absolutely. Modern genetic analysis, especially looking at ribosomal DNA sequences, strongly confirms they belong together as a distinct lineage. It's not just superficial, it's deep in their biology and their genes.
Right, solid evidence. Okay, you teased it earlier. Let's talk about this Straminipilous flagellum, the hair-like thing that gives the kingdom its name. You said it's an engineering marvel.
No, it absolutely is. It's fascinating. So first, maybe a quick reminder. A typical eukaryotic flagellum has that classic 9+2 arrangement of microtubules inside.
Right, the standard internal scaffolding.
Exactly. Now, in Straminipila, you often find two main types of flagella. There's the whiplash flagellum. It's relatively smooth, might have a fine coat, usually has a pointed tip.
And how does that one move the cell?
It generally pushes the organism through water. Think of like a simple propeller.
Okay, standard pusher type.
Mm-hmm.
But the Straminipilous one, the tinsel flagellum, sometimes called, that sounds different.
World's different. That's the star. The tinsel or Straminipilous flagellum. Imagine like a tiny feathered oar. It's covered with thousands of these really fine hair-like structures. They're only about 1 to 2 micrometers long.
Wow.
And they're called tripartite tubular hairs, or TTHs.
Tripartite tubular hairs, TTHs. Okay, what makes them tripartite?
They have this really intricate structure, a conical base that anchors them, then a long tubular shaft, which scientists think is made of 2 coiled fibers, and finally, loose, kind of splayed ends at the tip, where those 2 fibers separate.
That sounds incredibly complex for something so small. How do they even get these TTHs out of the flagellum?
Well, they're actually pre-assembled inside the cell in little sacs called Golgi-derived vesicles. While the zoospore is maturing, then these vesicles move to the edge of the cell and fuse with the outer membrane, essentially deploying the TTHs onto the flagellum's surface.
Wow. And what happens when the spore, decides to stop swimming and settle down? Insist.
Good question. If the flagellum gets withdrawn into the cell, those intricate TTHs are just shed, sloughed off.
Like discarding ores you don't need anymore.
Exactly. And they often remain behind as a distinct little tuft on the surface of the cyst. It's actually a helpful feature for identifying them.
So they leave their rowing equipment behind. Okay, the crucial question. How do these incredibly complex ores actually move the cell if they're not pushing like the whiplash type?
This is the really ingenious part. Unlike the whiplash flagella, the Straminipilous flagellum with all its TTHs typically pulls the spore through the water.
It pulls. How does that work?
It's quite remarkable. As the main flagellum undulates or beats, the TTHs kind of sweep backward, but the hydrodynamic effect is that they generate a force pulling the cell forward.
That's counterintuitive, but cool.
It is. There was a great analogy coined by Michael Dick back in 2001. He described it as being like a rowing 8 with fixed oars and a flexible keel.
A rowing 8 with fixed oars. I can picture that. The hairs are the oars fixed, and the flagellum is the flexible boat.
Precisely. And the sheer complexity of constructing this flagellum, this TTH system, it's such strong evidence that it probably evolved only once.
Makes sense.
So it's a really powerful evolutionary marker for the whole Straminipila kingdom.
Okay, moving on to our first specific group within Straminipila. The Hyphochytriomycota. Colloquially, hyphochytrids. You said it's a small phylum, but they've got something really distinctive.
They absolutely do. Their defining feature, their diagnostic characteristic, is truly unique in the known biological world. Their zoospore has a single anterior Straminipilous flagellum.
Just one. And it's the hairy pulling type at the front.
Exactly. Imagine a tiny swimmer being pulled along by just one of those feathered oars right at its nose. Nothing else known has that setup.
Wow. What else is notable about their spores?
Well, they also have that prominent Golgi stack we talked about and lipid droplets for energy. And intriguingly, they have something called a dormant kinetosome.
A dormant kinetosome? What's that?
It's basically the basal body, the anchor point where a second flagellum would normally grow from. but it's non-functional here.
So, like a leftover part.
It's interpreted as exactly that, the vestigial base of what was likely a whiplash flagellum in their ancestors. It suggests an evolutionary link to organisms that maybe had two flagella, the typical heterocont condition.
A little evolutionary echo. Very cool.
Yeah.
And biochemically.
Still, Straminipila, they use that DIP pathway for lysine synthesis, just like the Eumycota.
Okay, so they carry this biological fossil record. Where do these tiny solo swimmers actually live? What do they do?
They're pretty widespread. You find them in soil and in aquatic environments, both freshwater and marine.
And their ecological role.
Mostly they act as saprotrophs, so they're decomposers, breaking down dead organic stuff, recycling nutrients, really important background players.
But sometimes more than background players.
Sometimes, yeah. They can also be parasites. They might parasitize algae or even other fungal-like organisms.
Have they ever, made headlines?
Well, there was one rather striking report back in 1969. A species called Hyphochytriomycota was apparently responsible for a devastating epidemic in marine crayfish.
Really? Crayfish?
Yeah, now that specific scale of event hasn't really been observed widely since, but it's a potent reminder, isn't it? Even these seemingly obscure tiny organisms can potentially have a significant ecological impact.
Definitely keeps you humble about who's running the show sometimes. Okay, let's picture their life cycle. You mentioned no known sex life.
As far as we can reliably tell, nope. It seems to be primarily asexual. So those single flagellated zoospores swim around, find a good spot, and then they insist.
Right, they withdraw the flagellum.
Withdraw the flagellum, secrete a protective wall, and those TTHs get left scattered on the cyst surface.
Okay, cyst formed. Then what? How does it grow?
It germinates and develops its main body, the thallus, in one of 3 different ways. And these different development types are actually used to classify them.
3 distinct strategies from the cyst stage. Interesting. What are they?
Okay, first is holocarpic. Holo meaning whole. Here, the entire cyst just enlarges, and all of its internal contents convert directly into a new batch of zoospores.
So the whole thing becomes offspring, like a self-sacrificing sac.
Pretty much, yeah. Nothing of the original thallus remains. Second type, eucarpic monocentric. Eu meaning true, carpic for fruit or body, mono one, centric center.
Okay.
Here, the cyst puts out root-like structures, rhizoids, from one point. These anchor it and absorb food. Meanwhile, the main cyst body enlarges above those rhizoids, eventually becoming the spore-producing structure.
So anchored down with a distinct body part making spores.
Exactly. One center of growth and reproduction. And the third type.
Third is eucarpic polycentric. Poly meaning many.
Right. In this case, a more extensive, branching, almost hypha-like germ tube grows out from the cyst. It spreads out, forming a kind of network. And along this network, several spore-producing structures, zoospores, develop.
So multiple reproductive centers along a branching structure. More like what you might picture for some fungi.
A bit more like that, yes.
Yeah.
Though structurally still different, of course.
And then from all these types of thalli, new zoospores are released.
And the whole asexual cycle starts again. Fresh crop of single flagellated swimmers heads out.
Okay. Now for our second group, the Labyrinthulomycota. You said earlier, traditionally studied by mycologists, but they don't really resemble true fungi strongly. What makes them so unique?
Their distinctiveness hits you right away with the flagella. Unlike the hyphochytrids with their single flagellum, these guys exhibit heterocont flagellation.
Heterocont, meaning different whips.
Exactly. They possess both types. A straminipilous, tinsel flagellum, and a smooth whiplash flagellum, usually with that pointed tip. That 2 flagella combo is classic heterocont.
Okay, so 2 different flagellas, and internally,
they still have those characteristic tubular mitochondrial cristae, fitting them squarely in Straminipila.
But the really wild thing, the feature that gives them their common name,
yeah,
it's how they live, right? The slime nets.
Oh yeah, the slime nets. It sounds like sci-fi, but it's real. They live and produce these incredible slime nets. Imagine this delicate branching network of membrane tubes covering a surface.
Like a microscopic highway system.
Exactly like that. And within these tubes, their spindle-shaped cells glide back and forth. Sometimes incredibly fast. People have measured speeds up to 100 micrometers per second. Sorry, not millimeters, micrometers.
Wow, 100 micrometers a second. That's fast for a microbe. How do they move?
It seems to be driven by contractile proteins, probably actin-like, within the cells interacting with the net. And these nets are produced by special organelles at the cell surface called sigenogens or bothrosomes.
Sigenogens. Okay, so they secrete their own tracks.
They do. And here's another unique twist. The cells themselves inside the net have a unique cell wall made of Golgi-derived scales composed of L-galactose. This wall is located within the slime net, between the cell's own plasma membrane and the inner membrane of the net tube.
So they're living inside a self-made, membrane-bound tunnel system and have a special wall inside that tunnel. Very specialized. How do they eat?
They feed by osmotrophy, absorbing dissolved nutrients from outside, and those slime nets aren't just for travel.
They do more.
Yeah, they contain degradative enzymes. They can actually break down complex stuff like plant material or even lace, burst open other microbial cells nearby. The net is both transport and an external digestion system.
Multitasking slime. Okay, let's break down the two main orders here and their impacts. First, the Labyrinthulales. Genus Labyrinthula is key here.
Yes, Labyrinthula is the main player in the Labyrinthulales. You commonly find them living on marine plants, especially eelgrass, Zostera, and also cordgrass, Spartina.
Eelgrass. That rings a bell. What about reproduction?
They do both. Sexually, after meiosis, they form special spores called myospores. Typically, you get 8 heterocont zoospores from this process.
The two flagella type.
Right. And interestingly, these particular zoospores in Labyrinthula often have a pigmented eye spot, which is unusual for heterocont zoospores in this broader group.
An eye spot. To sense light.
Presumably, yeah. Asexually, their spindle cells can just divide within the slime net, and if a piece of the colony breaks off, it can start a new one. Very effective spreading.
Okay, now the eelgrass connection. This group has some ecological infamy, right?
Oh, absolutely. Labyrinthula species were identified as the pathogens behind the devastating eelgrass wasting epidemic back in the 1930s, primarily on the Atlantic coast of North America and Europe, actually, not just the West Coast.
Okay. Nice coast, primarily.
Oh.
And it was bad.
It was catastrophic. It wiped out huge percentages of eelgrass beds. Eelgrass is a foundational species in those coastal ecosystems, provides habitat, food, stabilizes sediment. Losing it caused massive disruption.
Impacting fisheries, too, I imagine.
Hugely. Fisheries that depended on the eelgrass ecosystem suffered major collateral damage. It was a massive ecological and economic blow.
Have there been repeats?
Not on that same devastating scale, thankfully. But Labyrinthula species are still commonly found associated with dying or stressed eelgrass. And then there's the more recent, surprising twist.
Go on.
A species named Labyrinthula terrestris turned up as the cause of a rapid blight disease on golf courses.
Golf courses. How'd it get there?
The thinking is it spread through contaminated irrigation water, possibly from ponds containing infected aquatic plants.
Wow. From vital marine ecosystems to pristine turf grass. It really shows how these seemingly obscure microbes can pop up and have major impacts in unexpected places, affecting industries like fisheries and golf course management.
It certainly does. You never know where the next challenge will emerge from.
Okay, let's shift to the second order, the Thraustochytriales. Where do we find these guys?
Thraustochytrids are also primarily marine, and they are everywhere, really ubiquitous. Found on decaying organic debris attached to surfaces, even living on the calcareous shells of things like mollusks.
And they're feeding on?
Organic matter, algae, bacteria, generalist decomposers, and consumers.
And their structure, still slime nets.
Yes, but it looks a bit different. Superficially, their thallus, their main body, often looks like a single spherical cell anchored by what look like rhizoids, kind of like some fungi.
But looks can be deceiving.
Exactly. Those aren't true rhizoids. They are actually the slime net, but in this case, it's typically produced by a single sagigen located at the base of the cell. So it forms this root-like attachment structure, but it is the slime net system connecting them to their Labyrinthulomycota relatives.
Clever disguise. What about their reproduction? Any sex known?
No known sexual reproduction for the Thraustochytrids. They release biflagellate heterocont zoospores, just like the Labyrinthulales. But these are mitospores. They're produced by mitosis, simple cell division, not meiosis like the myospores of Labyrinthula. That's a key biological difference.
Mitospores versus myospores.
Got it.
Do these spores have eye spots?
Generally, no obvious eye spot like in Labyrinthula. But interestingly, they are still phototropic. They react to light, particularly blue light, and they're chemotactic, meaning they can sense and swim towards chemical signals.
So even without an eye, they can navigate towards light and food.
Seems so. Very effective for finding new food sources in the water column or on surfaces.
Okay, and here's where this group gets really interesting from a completely different angle, right? Not ecology, but industry.
This is the major kicker for Thraustochytrids. Their industrial importance is huge and growing. Genera, like Thraustochytrium and Schizochytrium, they've become stars.
Stars? Why?
Because they are incredibly efficient producers of polyunsaturated fatty acids, PUFAs. especially omega-3 fatty acids like DHA and EPA.
The stuff everyone's told to get more of. Fish oil stuff.
Exactly that stuff. Vital for brain health, heart health, development. The list goes on. These marine microbes are basically little factories for making these valuable oils.
So instead of getting it from fish,
We can cultivate these Thraustochytrids in large fermenters and harvest the oils directly from them. It's seen as a potentially more sustainable, controllable, and vegetarian alternative to fish oils.
wow.
Thraustochytrid oils are already on the market in nutritional supplements, infant formula, animal feeds. It's a major application. They could genuinely compete with, or at least significantly supplement, fish oil production globally.
That's incredible. Talk about connecting microbiology directly to global health and economics. These slime net guys are punching way above their weight.
They really are. A fantastic example of how exploring biodiversity, even in these fungal imposter groups, can lead to really significant discoveries with tangible benefits.
So let's wrap this up. What does this deep dive into the Straminipila mean for you listening?
Well, hopefully it's shown that the world of fungi is maybe broader and weirder than you thought.
Definitely. We've seen this group, the Straminipila, stands totally separate evolutionarily, despite sometimes looking or acting like fungi.
Right. We've dug into their unique biological blueprint, those cellulose walls, the tubular mitochondria, the special Golgi, that amazing Straminipilous flagellum, the DMP pathway.
All setting them apart. And we saw their diverse lifestyles, from the single flagellated hyphochytrids acting as decomposers or parasites,
To the Labyrinthulomycota building those incredible slime nets.
and the real-world impacts are just staggering. Ecological devastation, like the eelgrass wasting disease.
And turf grass blight on golf courses.
Right. And then, on the flip side, becoming crucial producers of essential nutrients like PUFAs that impact global health and industry.
It really underscores how these microscopic details, cellulose versus chitin, tubular versus lamellate cristae, aren't just trivia. They define entire kingdoms. They dictate ecological roles.
And they determine whether an organism causes a blight or becomes a source for vital supplements.
Exactly. The Straminipila are a fantastic reminder that the natural world is just so much richer, more complex, and intricately connected than we often appreciate.
So the next time you think about fungi, maybe spare a thought for these fascinating, not quite fungi, the Straminipila. Remember their hidden but powerful influence.
This makes you wonder, doesn't it? What other unrecognized kingdoms or branches of life are out there quietly shaping our planet, shaping our lives in ways we haven't even begun to grasp yet?
A thought to ponder. Thank you for joining us on this deep dive. We really hope you've enjoyed this exploration into the amazing and maybe slightly mind-bending world of the Straminipila.
This has been The Deep Dive, brought to you by the Last Minute Lecture Team. We appreciate your curiosity.